Welcome, everyone. This afternoon, we're going to talk about tree anatomy, physiology, and the value of the Arborplug. So let's get started. My name is Joseph Doccola. I am Director of Research and Development for Arborjet. I've been that for the last 22 years. I've been in the tree and nursery business for I hate to admit, 40 years, and I am an ISA certified arborist. I'm also, I've got my horticultural certification through the New York Botanical Garden, and I'm a graduate of Cornell University. That's some of my background in case anyone's interested. So we recommend the use of Arborplugs, and the question is, why Arborplugs? And well, Arborplugs keep the injected product in the tree, for one thing. They ensure that the full dose is delivered, helps to prevent backflow or backsplash to the arborist, the applicator, reduces the risk to the cambium and possible consequential phytotoxicity to the vascular tissues. We'll get into a little more about that in a bit. It speeds up the closure of the injection site, and it will help to prevent secondary invasion of insects and disease. So what's the Arborplug? Said that it prevents backflow. It does so by an internal self-healing septum. And that is shown in the illustration here. It's a rubberized septum. The way it's constructed is kind of a dam. So as liquid is put into the through that Arborplug, any back pressure kind of pushes back on that semicircular part of the septum and seals it closed. So it is designed to reduce exposure to the applicator in the environment. The Arborplug facilitates systemic uptake, and the way it does that is via the infusion channels that it has around the legs of the plug, and those infusion channels, there's an illustration here in the bottom right, are these openings that allow the liquid to flow from the plug beyond the septum into the vascular tissue, into the xylem tissues. So those infusion legs are where the action is happening. It's where you have access to conductive tissues. The anchoring barbs, regard them sort of as a cork or a plug. It protects the lateral cambium from the injected chemistry. Sometimes these chemistries and the success of systemic injection has to do with chemistries being concentrated. So what you really want to do is protect that lateral cambium, and we'll look at where that is and what that means in a little bit. And it provides a surface for the injection site to close over. So the features of the Arborplug and the correct Arborplug placement. The Arborplug has 3 external and 1 internal component, which is the guide cap. It functions to guide the set tool on the exterior and an injection needle on the interior. Anchoring barbs, those function to create a seal between the sapwood and the cambium. Infusion legs allows liquid to flow in, and finally the internal septum prevents backflow out. So where do you inject into a tree? So in choosing injection sites, the idea is to inject at the base of the tree into the root flare or the trunk flare tissues. And what you really want to do is avoid the areas that are between those trunk flares. So what you want to do is find sites that are healthy around the base of the tree that are growing, that are actively growing. It may or may not be obvious, but you don't want to inject into a dead or decayed area or something that's physically injured. So you drill holes into the trunk or root flare of the tree. The reason for that is it favors canopy distribution. You let the roots decide where to distribute it the injected product into the canopy of the tree. It also favors injection site closure. And the reason it does that is because trees put down annual tissues. And if you look at it, the way I like to look at the construction of a tree is sort of like a stack of sugar cones, of ice cream cones. It's one layer over the other, and it's widest at the base. So most of the division and closure happens down at the base of the tree, and that is the reason I favor injecting low in the trees. Generally, you space holes around the tree, and these are basically deciduous trees, hardwoods, and conifers. It's a little different if you're injecting palms, but what I'm speaking mostly about are hardwood and coniferous trees. And generally you space holes 4 to 8 inches apart based on what your target is in the tree and based on the label indications of the product that you're putting into the tree. So a little bit about tree anatomy. So the way trees are constructed, they're highly compartmented organisms. The interior in a fairly mature tree has both sapwood and heartwood. You'll sometimes see sapwood referred to as xylem tissues. The exterior to that is the cambial tissues, and that cambium is occurs between the sapwood and the phloem. And that the cambial tissues are embryonic tissues. They're designed to actually divide and create more sapwood to the interior, more xylem to the interior, and to the exterior, more phloem. So these are initials. They're undifferentiated initials that create more tissues. Sapwood on the interior, phloem on to the exterior. The bark, what we refer to the bark or the cork, there's a separate meristematic tissue called the cork cambium that is beyond the phloem, and that actually makes cork tissues or bark tissues, or what we regard typically as bark. The heartwood is inactive in terms of flow in the tree. It's basically where the all the byproducts or waste products are deposited within the tree. It resides in the heartwood. It may be colored or not. The sapwood, actually, if you look at sapwood, it is a network of both these tubes, vessels, sometimes vessels and tracheids, but they're basically pipes within the tree that are much like straw. They're dead when functional. However, there's a reticulated network around that of live tissue. That web of live tissue is called parenchyma tissue. And parenchyma occurs both in the axial direction, that means with the bole of the tree, and it occurs radially, which means it goes toward the heartwood and out toward the phloem tissues. So this is injection of a dye. This is some of the early work that we did to look at Arborplug placement within the tree using dyes, and then subsequently autopsying the tree or cutting the tree to see how it functioned. And there's a couple of things that I would like you to notice. The sapwood is lighter in color, and let's see, it occurs in this region here. You can see my arrow. I hope you can. And the heartwood is the redder interior. What we did purposely here on the left is actually drilled into the heartwood material and then injected the dye. And what you'll notice are a couple of things here. In cross section, which is this image to the right, you'll see the dye flowing through the sapwood. And although it was drilled, we drilled a bit into the heartwood, you'll see very little movement in the heartwood. So heartwood tends to be plugged in trees, and sapwood, and this is what when we say active xylem or active sapwood, is open to movement of liquids in the tree. Generally, trees will move water and minerals, and that's from roots to shoots. The phloem is outside that area. It's kind of the inner bark, if you wish. And the cambium is a few cell layers between the sapwood and the phloem. And you can see that there's a region between between this cambium and the infusion that occurred that is clear. And that's where the anchoring barbs occurred to protect the cambial tissues. So this happened to be a dye, a water-soluble dye. It wouldn't hurt the cambium in any way, shape, or form. However, this illustrates that when systemic injections are done, it's the reason we describe a particular placement of that arbor plug into the tree. So if you knowing this is great, and that's fantastic. However, trees differ in their anatomy, and it's the reason I wanted to talk a little bit about anatomy and physiology here. There are three basic types of hardwood and coniferous trees, if you will. One type of vascular system is called ring porous, and the description of ring porous trees, and it's just this larger image to the upper right here, is you could actually count rings, and the reason you can count rings is you have larger springwood vessels forming before the smaller summer wood vessels, and because that alternates yearly, generally, you can count, we call rings, and rings occur when you cut a tree in cross section, but they're growth increments. So these are annual growth increments. And so some of the ring porous trees are ash, oak, and elms. They have relatively large vessels, and they are the fastest trees to inject and uptake, and they are faster in general than some of these diffuse porous trees. And I'm simplifying this. There are these semi-diffuse porous, but just to simplify it, it's these three areas that I'm talking about today: ring porous, diffuse, and non-porous trees. Some examples of diffuse porous trees are things like maple, beech, birch, and willow. And there's an image of a willow here. You can see it's it's the same kind of magnification, but much smaller. And generally, some of these diffuse porous trees, the reason they're called diffuse porous, because in cross section, it almost looks like these larger vessels are bubbling through the later vessels, so they're kind of intermixed. I know this is an oversimplification because a lot has to do with wood density characteristics as well, but that's kind of beyond what we're going to be talking about today. The third type are conifers or gymnosperms that rely only on small diameter tracheids. So tracheids are unlike vessels. They're not end to end. They kind of they're kind of weird. They kind of approach each other. If you put your two fingers together, instead of your fingers this way, you kind of go that way with them, if you can see that. And that's how they kind of attach. So they are kind of a slower event. Some of these parenchyma cells get together in groups and form resin ducts. In some conifers, pine and spruce have these resin ducts. And sometimes when you drill into a tree, you'll get resin flow, which is a wound, or basically a natural wound response in these trees. You won't see that in all trees. You won't see that, for example, in hemlock. Hemlock has resin ducts, but the resin ducts in hemlock strictly occur within the bark of the tree and not necessarily in the sapwood of the tree. So what does all this mean to systemic injection and the Arborplug technology that we're here to talk about? Essentially, if we were to look at, we're going to read this from kind of the right to the left. If we look at different types of trees with different vascular arrangements, and I picked oak as an example for a ring porous tree, sycamore for a diffuse, and hemlock as a non-porous tree. You'll see that if you look at the how large in micrometers some of these vessels or tracheids are, oaks would typically range anywhere between 50 to 200 microns across. And there's a formula on Uptake, and it was actually developed concurrently by both a German and a French scientist in 1839. That really says that the movement within a vessel is related to the fourth power of the radius, which means that the larger the radius, the faster the volumetric movement through that pipe or straw or vessel, if you will. So here you have the largest vessels or vascular tissues in oak up to 200 microns. You can actually use a stinger tip, and our stinger tip is not an Arborplug. It's the smallest diameter injection site. It relies on a 7/32 bit or a 5mm bit, so you can inject and go your merry way. It's going to uptake very efficiently. Sycamores, on the other hand, have a somewhat smaller vessel diameter and trach. It's anywhere from 25 to 100 micrometers. You can use a smaller Arborplug if you wish. The number 3 Arborplug, which is a 9/32 or 7mm diameter bit, is what you would use to put that in. And in hemlock, which is the most resistant because it has the smallest, and here I'm saying it's anywhere between 10 and 20 microns, sometimes as small as 3 microns. That's where you use the largest plug, and that requires a 3/8 bit or 9mm bit. So the idea here is the diameter is capturing a wider number of vessels or tracheids in that application. Now, can you use the Arborplug in all these cases, or the number 4 Arborplug in all these cases? Sure, but this is just the guideline if you chose to use not to use a plug. If you want to use a stinger tip, it's usually the most efficient in trees that have the largest diameter vessels. So let's look at what happens a little bit after the injection site. And these images are in ash trees. These are some of the autopsies or dissections that I did 4 years essentially after we injected trees in Lansing, Michigan. And these trees were attacked by emerald ash borer. And what we're seeing is that if you look at this line here, that is the point at which the injection was made. And I know this is pretty light, but there was at least 3 subsequent years to closure. And the reason there's 3 is this injection was done in the fall, and we had a complete closure. This is a radial view. This is the tangential view to look at the outside of the closed injection site. So I just want to say a little bit about how trees respond to injection site drilling. And it's a different strategy from animals. They developed a strategy of growing around the traumatized tissue rather than expending energy to repair the wound. So that wound will always be there throughout the life of the tree. The compartmentalization process that was advanced by Al Shigo quite a bit, and then other people have subsequently kind of modified it over time, but it's basically divided into 2 processes. And those 2 stages are the tissues that are in place at the time of the injury. You know, it could be an injection wound, it could be a pruning one, but it's those tissues that are present at the time of whatever procedure, whether an injection or a pruning wound. And then the second stage is really the responses or the growth that occurs after that event occurs. So, and reaction zones, you could also call them walls. they are dimensional. They have 3 dimensions. The fourth wall also has a temporal aspect, a time aspect, because it occurs afterwards. But basically axial, which means that it occurs with the main stem of the tree. It's vertical. So that is reaction zone 1. And the numbers here are not arbitrary. The least the slightest ability to compartment within the tree is the vertical through the vertical axis, because that's how trees move products. So it's least resistant to vertical limitation or limiting a wound in that dimension. The second reaction zone is inward toward the heartwood or the center. That's the radius, the radial is reaction zone 2. It is a stronger wall, and generally it occurs to the depth at which, in this case, if you're talking about injections, and that's what we are, to the depth at which you drill. And the third is the tangential or lateral movement, and it is the strongest wall. So that's reaction zone 3, is the strongest limiting wall. It kind of makes sense for trees. If you're a perennial and you did it the other way around, it'd be very easy to get girdled pretty quickly. But because of these reaction zones, because of how trees move products, and because of how they limit defect or decay or wounding within the tree, it survives. So it can tolerate these things very well. This happens to be an ash autopsy, and the red arrows are pointing to the width or reaction zone 3. It's very limiting, as opposed to the depth, the radial depth, which is longer. So that is actually the depth at which it was drilled. That's this image to the right. The image to the left was actually a stinger injection. With a stinger injection, you don't need to go as deep as with an Arborplug. Of course, the depth at which you do drill is based upon the skill of the applicator, but the width of the reaction zone 3, or the lateral movement, is actually very similar in both cases, but it's limited by the parenchyma, the radial and axial parenchyma in the tree, which are living tissues, undifferentiated, and are very important to tree actually wound responses as well. So this image here is, let me move this a little bit. You do that. Okay. So this is the radial section through the injection site with an Arborplug. Those arrows point to when the injection was made, and 2 years of new growth resulted in the encapsulation of that Arborplug. And wall 4 is this from those arrows out toward the bark, those 2 subsequent years, that's new wood being put down. That is after the injection was made. That's wall 4. And again, here's a tangential view of the exterior on the left of the wound closed. So how do you properly set the Arborplugs into the tree? Well, this is probably the most technical part of tree injection, because once you set the Arborplug, it's just inserting a needle and following the label directions on the amount to put in. And so what we really want to do here is protect the cambial tissues. So what we basically advise, or what I'd like to advise, is when you drill into the tree, just drill first. The length of the Arborplug is it's about 5/8 of an inch. So you have to drill 5/8 plus the thickness of the bark. Bark generally, it has a lower density than sapwood. So if you were to first use a cordless drill and just simply squeeze the trigger without pushing, often enough that drill is going to land, it's going to stop going in at the point where it meets the sapwood. So just remove the drill and just check out how deep that bark is. that will give you an indication of the countersink, how far you have to sink the arbor plug in using the plug setter. And if you do this, you have a nice surface to inject. You could kind of rest assured you're not really going to expose the cambium to any concentrated liquid of the injection material that you're putting through the tree, and it's going to move very efficiently in the tree column and distribute well in the tree. So this is probably the most technical aspect. It's okay, let's get that arbor plug set and countersunk in the tree. You'll get good movement within the tree. And here's an example 2 years after an injection. The arbor plug is really encapsulated well within the tree. You can see around it, this wall 3 is very strong. There's very little lateral movement around this drilled site. So that's the strongest wall. And the tree put out new growth after the injection was completed. And again, this was an ash that was attacked by emerald ash borer. It survived it until I decided to autopsy it. In this case, you can see that at the time of injection, the rings can be reduced in thickness, and that is an indication that that tree was undergoing stress and attack, in this case by emerald ash borer, and then subsequent to an injection treatment we used in this case triage. You can see the width of these rings have recovered. You can see closure around the injection site. There was some included bark within that, but it subsequently also closed over. So kind of a nice success story there. This is just a cross section, whereas the previous image was a radial section showing the 4 years of subsequent growth following in injections. This section is a transverse or a cross section that is above the injection points. And finally, what we want to look at is putting this all together, what does that process look like when we approach tree injection? Generally, much of the label indications say, hey, listen, measure the tree and let's look at the diameter at breast height, which is 54 inches above the soil level. And generally, we use either DBH divided by 2 or DBH divided by 3, depending on the technology you're using, whether it's the QuickJet Air or the F series tree IV. So that drives that. Generally look at the label or give us a call for that advice. But you're choosing the injection sites. These red circles indicate low in the tree, which is correct. You drill, again, you first drill into the tree, apply no pressure, look at the bark thickness, go in, drill to at least 5/8 of an inch in depth. In most trees, you may want to go in conifers because they're very resistant to injection, to go a little bit deeper than that. Place the plug in the hole, tap the plug into the correct depth, you're countersinking it into the bark, and then you inject through the plug. So that's basically the methodology. There are some factors that affect how efficiently trees will uptake the injected solutions. The thing that I think really drives this primarily is soil moisture. So transpiration favors uptake. And if you have soil moisture and low relative humidity around the leaf of the tree, injection is favored. So uptake is favored under those conditions. Humidity, relatively low ambient humidity favors injection uptake. Temps, you want soil temperatures, you want available moisture, so that the soil temperatures have to be above freezing. So they have to be above 32 degrees, but ambient temperatures should be less than 90 degrees when you're doing injections. A light wind will favor evapotranspiration and uptake. The wood vascular characteristics we talked a little bit about. Trees with large vessels and relatively low wood density favor uptake. Note that softwoods rely solely on tracheids, which restrict or are slow in uptake. Tree health and vigor, intact vascular tissues, and wide growth rings reflective of twig growth favor uptake. Injection formulation. Formulations that are designed specifically for tree injection uptake more efficiently. It's basically the material, and it's based on tree physiology. What do trees move? They move water and dissolve solutes. The material that you put in through the tree has to mimic what trees naturally move for efficient uptake. And the applicator skill, and most of that has to be about proper plug set. So what are factors that don't favor uptake? Well, drought, excessive rainfall, ponded soils may reduce uptake. High humidity, if there is, if the canopy is not losing moisture efficiently, and evapotranspiration from the canopy slows, uptake slows. Temperature extremes. If the soil is frozen, there's no available water. If it's too hot, you can possibly get leaf injury and leaf senescence in the fall. Sorry. Wind. Excessive wind may close leaf stomata. Light winds favors evapotranspiration. Wood vascular characteristics, mostly tracheid types, which are your conifers, especially your resinous conifers, may be barriers to uptake. High wood density. One of my old professors actually has a short document in what was then Journal of Arboriculture, and it was wood densities in reference to injectability, and that was Wayne Sinclair. Physical tree issues, if there are girdling roots, injuries to the bark, if the canopy was, if the tree was excessively pruned or topped. I mean, these things do not favor injection. Diseases, insects, defoliation, those kinds of things. Remember, trees are plants that are autotrophic, which means that they have to make their own food. If they're hamstrung at that, they're not going to work efficiently. So when you approach a tree for, if you want to do systemic injections, approach the trees that are at risk of an insect or disease based on what's occurring around that tree, but try to be proactive about it, because it's really difficult to fix what's broken, especially with things like emerald ash borer, which causes vascular mining under the bark. You don't see it, but if you get in there too late, you're not going to get a very good result. So that may be a barrier to tree recovery. So just keep that in mind. Formulations, the products that are not designed for tree injection may inject poorly, have reduced efficacy, and may cause undue phytotoxicity to tree tissues. And applicator skill. And this, I don't want to underemphasize. The plug set, if it's shallow in particular, you can cause injury to the cambium. Pressurized injections, which are most of our technologies, could cause physical cracking if done improperly. So, and that's the reason here today to talk a little bit about proper plug set and methodology. And let's see. And that is pretty much it for today.